The last macromolecule we're going to talk about are the nucleic acids, but this is the shortest and sweetest of the four because we cover this in greater detail when we get to the genetics chapter. Genetics is all about DNA and RNA, so nucleic acids is pretty much your DNA. So let's take a look at their general structure. First of all, the elements that it contains is everything except for sulfur. So of schnapps, it's just schnapp. So no sulfur in DNA or any nucleic acid. Their main function is because it's your genetic information. So they could store and transmit genetic information. And they do that in a process called protein synthesis. But wait, proteins was another macromolecule. Yes, they are actually the instructions for why you get all the proteins that you do. And that's a whole nother chapter. So they will determine how proteins are made and their sequences. Remember, the sequence of a protein is what amino acid of the 20 is going to be next. And that's all determined by your genetic material, by your DNA. Now, DNA and RNA are considered the polymers of nucleic acids. So I think that's on your note sheet, that the two different polymers are DNA and RNA. And we're going to look at these closer up later in the year, but a little bit right now. Now, when we talk about it determines protein sequences, later in the year we're going to do a chapter called protein synthesis, and that's how proteins are made. This is kind of a very brief overview right here. So we're going to take our DNA. Our DNA is found in every cell in our body. There is an exception to that, but it's found in every nucleus. It's found in the nucleus of every cell of our body, and it's the instructions for how to make stuff. The stuff that it's instructions for is how to make proteins, and proteins make up your body. And so the DNA is too important to leave the cell. And so it will send a little messenger, a little photocopy of itself out in short little segments called RNA. And the RNA actually is read as a sort of instruction book for how to make the proteins. And so this picture is showing in the yellow DNA, and then this white strand that's coming out is the RNA. The RNA goes out into the cell, and the cell, you have cell structures inside of there. They're called ribosomes. They will actually read the RNA, and they will actually make the proteins. So each one of these circles are actually amino acids. And there's the peptide bond that's holding them all together. And down here, you could see there's this folding structure of the proteins that we talked about the other day, how they twist in the fold. So this process is called protein synthesis. You go from DNA to RNA, and RNA to the actual protein. And so it should all make sense by the end of the year if it doesn't right now. Let's look at the monomer of a nucleic acid. So the monomer of a nucleic acid is called a nucleotide. Nucleotide, here's the structure of it right there. You have this on your picture, on your paper notes, so make sure that you fill out what each of them is. So the first one is you want to find the circle. The circle right inside, that it is the phosphate group. It's abbreviated PO4. There's 4 oxygens to the 1 phosphate. It will never change. It always remains the same. The second part of a nucleotide is actually the sugar. The sugar is usually a pentose. Remember, pent means 5, os means sugar. Goes back to the carbohydrate. It means a 5-sided sugar. And you could see that this sugar definitely has 5 sides. So sometimes we call it a 5-carbon sugar instead, because there's 5 carbons in that ring-like structure. Now this can change. It depends on which one you have, DNA or RNA, so we're going to talk about that. The last part of a nucleotide is called the nitrogenous base. Sometimes we just shorten it to nitrogen base. Sometimes we shorten it even further to just nitrogen, or to just the word base. But right now, let's keep it nitrogenous base. That's a fancy word for meaning it has nitrogen in it, and so you can see all the nitrogen that you find inside of it. This one can change in actually 5 different ways. So we actually have 5 different versions of what could go here. This whole structure right here is called a nucleotide. The phosphorus or the phosphate never changes, but the sugar and the nitrogen bases can change. It depends if you're talking about DNA or RNA. So if you're talking about DNA, in DNA, here's a DNA nucleotide. You have the phosphate group. Okay. Here's RNA. RNA, well, by the way, DNA stands for deoxyribonucleic acid. 2 words, really long. But deoxyribonucleic acid. And so DNA is right here in DNA. That's where it gets its name from. But DNA has a phosphate group in its nucleotides. Okay. Here's RNA. It stands for ribonucleic acid. There's the phosphate group still. So they're the same. They do not change. The things that change are right here. So here's the differences. In a DNA nucleotide, you actually have a sugar called deoxyribose. We talked about that. It's a monosaccharide. So deoxyribose, it's a pentose, 5-sided sugar. In RNA, you have a slightly different sugar called ribose. Very similar. It's a 5-sided sugar. The only difference between the two is right here, and it's highlighted. So this is the only place that it's different. It's deoxy because deoxyribonucleic acid, DNA, has one less oxygen than the ribonucleic acid, than RNA. So it's only missing one thing right there, but it makes a big difference on whether or not it's a DNA or RNA. Then the last thing is the base. So this base right here, okay, it's representing in the gray right here. There's actually 4 different versions of what it could be if you're talking about DNA or RNA. This base could be a T, an A, a C, or a G. It stands for thymine, adenine, cytosine, or guanine. One of those 4 could be in this place. So there's actually 4 different versions of nucleotides for DNA. In RNA, the bases could be 4 different things. They could be U, A, C, or G. Uracil, adenine, cytosine, or guanine. So one of those 4 could be up here. So there could be 4 different versions here. Total, there's 5 different letters. The only difference between DNA and RNA are these 3 things. We have a deoxyribose sugar. Here's a ribose sugar. If it's DNA, DNAs have T's for one of the bases called thymine, but RNA, those are replaced by U's, which are uracils. The adenine, cytosine, and guanine, A's, C's, and G's all stay together. The last difference between DNA and RNA is that structure determines if it's double-stranded or if it's single-stranded. And you could see, here's a nucleotide, here's a nucleotide, here's a nucleotide, and here's a nucleotide. You put a bunch of monomers together, you get a polymer, and we call this polymer DNA. And you can see they're actually attached to other ones going down the other side in the middle, where the bases are. Now, RNA doesn't like to attach to anybody. It's single-stranded, but you could see that here's one here's one nucleotide, 2 nucleotides, 3 nucleotides, 4 nucleotides, and this one is called RNA. So DNA and RNA are polymers of nucleic acids. Looking a little bit closer at the bases, so we're going to look at this when we get to DNA and RNA, but the bases are slightly different. So in DNA, you could have T's, A's, C's, and G's. These 2 will always bond together, and these 2 will always bond together. That will make more sense later. And then in RNA, you have U's instead of the T's, A's, C's, and G's. And these 2 always go together, and these 2 always go together. And this is a DNA nucleotide, and this one is an RNA nucleotide instead. So, I think the last thing on your notes is saying that if it contains a deoxyribose sugar, it's going to be DNA, but if it contains a ribose sugar, it's going to be RNA. This down up here, this is a picture of DNA, and this is a picture of RNA. One's double-stranded, one's single-stranded. And these are the bases that make each of them up, along with the different sugars. So that's pretty much all that we're going to talk about. We'll talk about a little more details in class, and then later on in the year. Okay, I'll see you in class. Bye.